What we're trying to understand at the broadest level is how genes influence behavior, both in mice and rats, which is what's most germane to what we're talking about today, but also in humans. And in particular, we'd like to extrapolate from the human data and bring that to the mice, and also from the mice and the rats and bring that to the humans. In 2014, we got a P50 Center grant from the National Institute for Drug Abuse. And in that project, we're looking at about 5,000 rats, which are really deeply phenotyped behaviorally, so that we know a lot about the behavior of each of these 5,000 animals. And then we take DNA from those animals and genotype them at hundreds of thousands to millions of polymorphic sites throughout their genomes, so that we can look for statistical associations between the genotypes and the phenotypes of the animals. Critical to our project is identifying genetic differences from animal to animal that are correlated with and that may explain the behavioral differences. So we need to have extremely pure, high-quality DNA, which we then subject to a sequencing-based genotyping technique. If the DNA is not of the highest quality and purity, the genotyping results that we get are not optimal, and we may lose or miss some of the correlations that we're interested in discovering.
We needed to choose the method, and at that point, two types of methods were available. Basically, a silica membrane-based method and beads method. Both methods allowed a 96-well plate format, which is great for scaling. Right, because you can barcode the plate and then you don't care about identifying individual samples. So we tried silica membrane-based method and it didn't work very consistently. Some of the samples worked, some didn't, and we wanted the method which will work almost 100%. So we decided to look at the beads methods and we compared several of them, and we definitely wanted the plate format as opposite to individual tubes. Ultimately, we selected AgentCore DNAdvance. It produced robust results, so almost 100% of samples worked every time.
It's very fast. You can do it in a couple of hours with one or two plates. And it is also very high throughput. You're using plates, so that's 96 samples in one plate. And we're getting really high quality DNA that's large fragments and very clean and very good for our genotyping by sequencing library prep.
So we've now phenotyped about 4,000 of the 5,000 rats that we will ultimately phenotype, and we've collected DNA from more than 3,300 of those animals, so that we're starting to see really robust statistical associations between particular regions that contain particular gene variants and the behaviors that we've observed in those animals. And then we can also stitch together statistical associations between gene expression in critical brain regions and those behaviors. Ultimately, so that we can identify chromosomal regions that both influence behaviors and influence gene expression traits, and we can then later test the hypothesis that the region is important for the behavior because it influences that gene expression trait, and that it's the gene expression trait that's influencing the behavior.
And then we hope that it will lead to some new clinical approaches which can be used for humans when the genes responsible for drug abuse behaviors are known.